I'm all caught up with returning things to, well, except for you, returning things to people. Now, so. For those of you who are here, I think I'm caught up now in returning your stuff. Let me know if I've made any errors in that respect. Okay. We'll talk briefly about the exam coming up, and then we'll begin the chapter on T-cell activation. I know it's always awkward having a lecture on stuff that won't be on the exam that's coming up, because people might not be as attuned to it, but I hope you'll find it interesting enough to do that. The other item is the papers. So I think everyone's been informed what you'll be doing, and we'll kind of get into depth of that, and let me know as problems arise. You know, I haven't looked at things really closely, but I'm going to vow to do that. Again, I picked these with the idea that they would be both new and topical and interesting to me, and I hope interesting to you as well. So I'm going to, yes?
So expanding on the journal club, what are the expectations? It's a half an hour presentation.
Well, I would think maybe a little less than an hour, I don't know. I hesitate to have a strict rule like that. I think I intended to post something. Let's see if I actually remember to do that. I thought I had posted a document, and I'm going to take the time to do that now. I think I'll go to the hints on the exam, and these may not be as illuminating as last time. It says, please wait. I'm waiting. There we go. So I don't have as many distinct hints, but I think, hope they'll still be useful. So DNA rearrangement, so just know really what goes on, what the initial arrangement is, and then the final product is. You know, those things. Structure and function of MHC. Structure, what proteins look like, and function, what they do. And none of the details, not every little thing, but why they do what they do for these key experiments. And I do them by name. You know, I don't so much think that one needs to worship great scientists or decide this person is better than that person, but it's easier for me to remember experiments if I remember by a name, okay? You know, especially if it's a funny name like Zinkernagle, right? So I don't believe in so much the, I'll say the worship of you know, the great people, but still, you know, one can remember, okay, when you say associate a certain person with a certain experiment, I think that's easier to remember. So I'm going to say no main points of these experiments and how they did what they did. And they're well described in the book, and I like what the way the book gives you original experiments, not the original papers, but describes well. I think they do an excellent job of that, summarizing things. As a teacher, I know that when you want to summarize something lengthy in a few sentences, it's difficult to get everything important, be completely accurate, and I think the Kubi authors do an excellent job of that, especially in these things. Okay, so questions on that line? Okay, so yeah, it might be a little more experiment oriented this time. Other thing, so as far as presentations, I prepared a document a couple of years ago as a guide. Now you've been doing these things for multiple classes now, and these are just kind of hints. The main thing is I don't want this to be like full of stress. I don't want it to be trying to, I think I have the words in there, don't trash the paper or trash the presenter. I don't know if you've been in situations like that with peers, but again, what's my analogy? Maybe we're going through the uncharted forest and the person presenting is the leader, bushwhacking through the forest, okay? Doesn't mean you have to know everything, but you've thought about it more. And it's okay to say, I don't really see how they do this, because honestly, for me, going through these papers, and I've been reading papers for a long time, I admit there are things that I don't understand. Well, certainly I don't understand at first, and then they even struggle with after a while. And I think probably most people will struggle with some things, with some techniques. Especially papers that have such huge amounts of data. So this is something that I prepared a while ago. So that's a link here. So here's some suggestions. And I decided what I would like, this would be my ideal, what I would like kind of tone as much as specific things. Okay, I did like some background, like what's been done before, where was the field before, and I have italicized like perhaps sample statements. I think this might have reflected papers that we might have read this one year, but, and here's my sample. People have been interested in allergy for a long time, but for technical reasons could not purify IgE producing cells. Okay. Mouse models have suggested that TNF has an essential role in this disease. However, this had not been directly shown in humans. Previous work had been hampered by a lack of useful reagents. There was no antibody specific for this cell type. Anyway, those are kind of phrases that I would find appropriate. Paper itself. In introduction, what is new in this study, their approach, their reagents, their conclusions. You know, I put down this group used new 30 color facts, and I put joke, you know, maybe that's not really a joke so much anymore, with many color facts to show that XXX cells alone could transfer disease. And since the work of, I made up names, but Guliaboff and Smith, everyone had thought that the interaction between CD40 and CD40 ligand was necessary for maturation of these cells. In our paper today, these authors suggest that the earlier conclusion may not apply in all cases. Okay. You know, there's a phrase, this applies to you guys who'll be doing presentations soon. Tell them what you're going to tell them, tell them that, and then tell them what you told them. Okay, and that applies. Presenting each figure. What are they trying to prove? Did they prove it? What more would you like to see? Explain the techniques should be used in the figure. Okay, and here's my simple phrase. The data shown is convincing. However, I would like to see a data point that goes out more than 7 days. See, that's one thing though. I don't want you to trash papers, but when you're the person commenting, you can demand great things of them. You know, go on a long time. You know, use more advice because it's not your time or your expense. Be critical of papers. This doesn't mean you need to trash papers. Okay, just say I'm not sure how you can say this, you know, because such and such. Sum up the paper. Do the conclusions fit the data? Are there other approaches? What should, and that's really a high degree of sophistication, recognizing different approach. You know, these guys did this in this mouse model, other people did it in a human model, things like that. And you know, are there different ways to do it? Did other people already do it a different way and get a different result? What should workers do next? So here in my phrase, they did a good job of showing that EAE, a mouse disease, is mediated by this cell type. It is unclear if MS, the human disease, is affected the same way. Okay? So those are, I don't know if that's helpful, but those are kind of like good characteristic sentences, perhaps. Okay. I hesitate to mandate a specific amount of time, but I would expect these might take an hour, maybe a little less. Okay, a little less than an hour, but that's a sample. And what people will roughly divide up the work and do give a background, like why they do a certain approach and then sum up. If something is not readily understandable, talk it over with your partners and come to me ahead of the date. Some of the techniques and data will be difficult to follow. This is how you learn, smiley face, okay? Easy thing to say, but yeah, and I need memo to self. I need to really go over these things because I need to be able to answer them when you come. So these are all immunity papers that I think were in press when I started looking. Not even, they're out in the hard copy journal yet. So we're going to be doing things that are good. I was decided to work backwards time-wise to have recent things, and I thought if I found something that was interesting enough, as far as big questions, and we could sink our teeth into data and the approach, then it would be good. So I found 3 pretty quickly. Okay. So I hope this goes well. I hope it'll be a fun exploration of topics that are new to you, rather than something that's too much of a burden. And I recognize, half of immunology is the names, the nomenclature, rather than what the experiment itself is. So, I hope that is useful as a guide. Yes.
And the dates. So we have 2 dates designated.
Oh, yes, now, so that means we could have 2 people, I think we'll have 2 groups present on one of the days and 1 group on the other day. And probably everyone will want to be later, right? If you have any compelling reason why you have to be one or the other, please let me know, but, so I haven't set that yet, but it'll be 2, I set aside 2 dates. Originally, I was gonna do 4 papers with groups of 2 or 3, but I decided to do 3 groups of 3 each. Okay, that's how we have that. Yeah, the person that goes alone, they can go for 2 hours. Just kidding. Already, any questions come to mind now? But important is to to dig into what's right there, but also context, because what's been known before, and they're usually pretty good about that. Saying that we're so great. This is the first demonstration. Although I know reviewers who will reject statements of what's the word, primacy? Is that the word? Who don't like that, but you can usually follow the field. If it's a good introduction, they'll say so-and-so did this, and so did this, so-and-so did this. Here, we describe the next step. So you're not supposed to say, we are the first to do this great thing, but you lead up to it and then let people conclude you are the first to do the great thing, okay? Anyway, so that's where we are. Okay, and so you have 2 weeks now, right? Correct, yeah. So I think that'll be okay. And I'm gonna vow to go over those things carefully. So now, today on chapter 10, we've gone over the development of T cells as well as B cells. So now they are, they've made it through selection. They're ready to go, or almost ready to go. So we're gonna talk about T cell activation. And the first, and there are many fates for our T cells, especially the CD4 T cells. And the first question that's raised is this idea of a second signal, and also referred to as co-stimulation. So we talked about our big picture questions in immunology: creation of diversity, making an adequate response, and avoiding self-reactivity. So ponder by yourself or with your neighbor for a few minutes about what the value of a second signal, and whether you've read the chapter or not, what the value of a second signal could be, what it is, and what the function of it is. So I'll give you a few minutes to do that. people ready to express their thoughts, musings? So what's the value of co-stimulation or benefit if co-stimulation is needed?
I think it's really critical not to sort of flippantly trigger the immune system. So having the requirement of multiple signals means that you're going to be really sure that it's time.
I think that's fair. So a break on possible inappropriate activation. Is there anything you want to add to that? That's good for summing that up. And it's amazing when you look at everything about the immune system, the development of the diversity and activation, but also all these checks. It's just remarkable. Yeah, because if you want to have that rare cell that escapes thymic negative selection, you don't want it going crazy in the pancreas and destroying the insulin-producing cells. And one way of doing that is saying that it's only going to be fully a CD8 T cell that might recognize insulin-producing cells if it's only activated or fully activated by an appropriate antigen-presenting cell as opposed to a pancreas cell.
I think also too, you don't want to distract the immune system and like allow something else to come in while you're attacking something that doesn't need to be attacked.
I think that's reasonable too. Yeah. And also, when we see the negative co-stimulations or the inhibitory co-stimulations that you don't want, you mount a huge response to some infection, and it would be bad if you had but you want to dampen that, and you don't want to have that continuing too long or those cells accumulating. So all those things. Okay. So objectives in this chapter: signals to activate, co-stimulatory signals, co-inhibitory signals, extracellular influences, including cytokines and different lineages, different helper T cell subsets, memory subsets. So the initiating interaction for response is between the antigen-presenting cell, or APC, and T cells, and naive T cells. So T cells are naive. Great usage of language, isn't it? Naive until they meet antigen, and they must be activated before they can perform their effector functions. They circulate in the body, browsing for antigen. Isn't that a great line? The T cells are browsing. What are you doing? Well, I'm browsing for antigen. So these B cells and T cells unactivated are kind of boring. They're small, not very active. So once encountered, T cell stops migrating, initiates a response. And as you guys know, you guys who study signaling, you know that the resting lymphocyte is a model system for something that's not doing anything, and then you can measure activation when you're just looking at a response to signal and response. So T cell stops migrating, initiates a response, and is no longer naive, which does not describe a naive T cell. Remaining stationary. Okay, so giving away the answer before the experiment, T cell activation, they say requires 3 signals. Classically, people talked about 2 signals, but they're saying no, 3 signals. So antigen presentation is the first signal. It is important that that's the signal that is specific or clonotypic for a given T cell. The other signals are not clonotypic, they're general and apply to a number of cells. So other interactions provide second required activation signal, and then cytokines providing the third signal. And activation allows differentiation, CD8 T cells can become killers. They're primed is the word often used. And then CD4 T cells can differentiate into several different subsets. And people, there was an initial amazement that they could define two subsets, Th1 and Th2, and it seems like the harder they look now, they find more subsets. But it's important to get the basic picture right. So naive T cells circulate among lymph node secondary lymphoid tissue in the blood. It's estimated that 1 in 10 to the 5th T cells are specific for an antigen and that they recirculate every 12 to 24 hours. So if the T cell encounters cognate antigen, people use that term cognate, the right match, the right fit. There is an immune synapse. So synapse is a word used in neurobiology. You see it more and more in immunology now. The affinity, just if you measure the TCR to MHC peptide affinity, is not very high. Caved associations from 10 to the -4 to 10 to the -7. But remember, it's not the physical chemist's experiment in a tube. It's cells with many molecules on them. So it's helped by avidity of the co-receptors, CD4 and CD8, and also by other molecules on the surface of those cells. You see here a cool figure here. So for full activation, there is needed interaction between CD28 on the T cell and CD80 or 86, formerly called B7-1 and B7-2 on the antigen presenting cell, especially the dendritic cell. So this was first called the two-signal hypothesis, and then it's now expanded to 3 due to the importance of local cytokines. And again, when we talk about local cytokines, another reminder that it's not really the pure biophysical experiment of affinity of one molecule for another. There's a local concentration of cytokines which are going to be higher than the overall concentration in the blood, or even might even be one side of a cell versus the other. It could be a different local concentration. So we see the overview here. APC could interact with a CD4 or a CD8 cell, and here the CD4 is activated, making cytokines including IL-2, which stimulates itself and others. We see different local concentrations there, clonal expansion of that CD4 cell. Or the CD8 cell can be activated. In each case, clonal expansion, and these can eventually become effectors, making appropriate cytokines. And you can see different effectors lead to activation of other cells, helper T cells being helpers. And the effector CD8 killer T cells killing target cells. A fraction of these will also become memory cells, which when called upon will make more effectors. The figure here in our lymphoid organ here. So optimal activation and proliferation only comes when there are co-stimulatory signals. Again, the co-stimulatory interaction is a general one between molecules that are on the surface of many cells, be it presenting cells or T cells. Signal 1, antigen-specific TCR engagement, that is very specific. Signal 2, receptors on T cells, mentioned CD28 and ICOS, and then the co-stimulatory ligands, CD80 and 86, ICOS ligands. And then signal 3, cytokines being involved in guiding T cell differentiation into different types. So we see in this figure, we see the TCR signaling, that's the peptide, the ball in the middle, the co-stimulatory interaction, and autocrine and paracrine signaling. And these are 1, 2, and 3. So there's been a lot of work lately on these synapses, again, that it's, these aren't just all evenly distributed on the cell surface. And look at this pretty picture here where they have staining for molecules, and they have the central area and then the peripheral area for the synapse. And in the center, TCR and CD3, the co-receptors CD4 and CD8, and CD28, and then these other adhesion molecules which help in the interaction on the periphery, forming this ring here seen in red. So that's kind of cool. So they call them central and supramolecular. Yes. So that's a pretty cool picture there showing that. And then our figure here with CD4 and CD8 cells and the interaction with the central. So an important point that there is the specific interaction between the TCR and MHC peptide, but also these other interactions between proteins that are not specific. But are very important. So the heading in the book says TCR signaling provides signal 1 and sets the stage for T cell activation. And there's a phrase, oh, I didn't check this, I have this in my notes from the last edition. I assume this phrase was not removed from the current edition because it's so good. It says, signaling cascades can be at once daunting and dull to study. So I think I really like that because that's certainly how I feel. Daunting, but dull to study. So that's a quote. Another quote in there, signaling is all about location, location, location. Because part of it is these molecules coming together and making a scaffold that's built.
Yeah, they're both still in the book.
They're still there? Okay, good. Well, I couldn't throw those up. I hadn't checked that for sure. Yeah, those are great. I feel, I express my feelings too for both of those. So early on begins with the tyrosine kinase, LCK. So this is the kind of thing that you've studied in cell biology before. LCK phosphorylates the ITAMs on CD3, and they become docking sites for ZAP70. LCK phosphorylates ZAP70, and we have this cascade here. So I'm not going to ask that you know all these for this class. You probably had some exposure to them earlier. So LCK, you may have heard about this from SARC family kinases before. LCK is activated actually begins with a dephosphorylation. You familiar with that? Kind of, yeah. So that's done by CD45, which is a phosphatase, and that changes the conformation of LCK and activates LCK. So once the ITAMs are phosphorylated, now proteins with SH2 domains combine. ZAP70 is a T cell specific kinase, which gets phosphorylated and is itself now active. Adapters LAT and SLP76 are substrates. So the book, I think, does a good job. It stresses architecture, physical relationships, scaffolds, I use that word a lot, are important. Sometimes there are elements present in one cell type, for instance, T cells, but not in another, like B cells, for instance. So in one path, phospholipase C gamma binds and removes the head from lipid tails, making IP3 and diacylglycerol, leads to release of calcium stores, activates calcineurin, which dephosphorylates NFAT. Remember, NFAT is one of the big transcription factors that's downstream activation to turn on genes. Diacylglycerol, DAG, leads to NF-kappa B activation. Also, GRB2 is an adapter which activates the RAS pathway. The MAP kinases phosphorylase serine. RAF, MEK, and ERK are among these. So, small number of interactions are needed for survival, proliferation, and differentiation. Lots of stuff here. So remember the use of LCK, tyrosine phosphorylation in scaffolds being important. Some of these things here. Again, notice the 3 transcription factors that are important in point activation of these lymphocytes: AP1, NF-kappa B, and NFAT. Now, signal 2, let's talk about what signal 2 is and what the evidence is for signal 2. So these are co-stimulatory positive receptors. They're also co-inhibitory receptors, including CTLA4 and PD1. So they can be called either negative co-stimulatory receptors or co-inhibitory receptors. And their role seems to be to maintain tolerance and reducing inflammation. So early on, people found that signal 1 without signal 2 leads to anergy. I want to be careful about the use of energy, because it seems to me back when I was attending seminars that people use energy in multiple ways. I think everyone would agree that there was some form of energy, meaning not responsive, not giving a signal there, but people might differ as far as their definition and whether they think energy meaning not being responsive in the future, whether that is what they're talking about with the energy. So the model is a signal 1 without signal 2 leads to energy. So this was initially done with planar lipid membranes, and this is one of those things that was not. They did not continue. This is a figure in the previous book that didn't continue here. So this is an experiment done by Carl June, and it's done looking at activation of T cells in kind of a very in vitro system, and the readout is treated uridine incorporation. Okay, so that would be like RNA production, RNA taken up and incorporated into macromolecules, an RNA precursor. Okay, and the way they did this is with planar artificial membranes, and they found that, bottom line is they found that signal through both the T cell receptor and CD28 was necessary for optimal signal. So here we have a figure here, we see the T cell receptor. Now, antibodies are tricky as far as their use in experiments. So antibodies can be used to block an interaction, a normal ligand protein interaction, but also antibodies themselves can lead to activation, especially if they lead to clustering of a given receptor. So anti-CD3, remember CD3 is a collection of proteins that are monomorphic, that are associated with the T cell receptor. So anti-CD3 can be used as a polyclonal activator of T cells, okay? And that was widely used, especially before people had the ability to work with you know, T cell clones and specific T cells, say in transgenics. So you can take a population of a wide variety of T cells with many specificities and treat them with anti-CD3, okay, especially on a surface, and that will cluster the CD3 and TCR and turn it on, okay? Now, similarly, they used anti-CD28, and their results here, that anti-CD3 alone gave a good signal, but that was much increased. Now, anti-CD28 alone gave kind of minimal signal, but anti-CD3 plus anti-CD28 gave a much better signal. So this was kind of the grandfather experiment of co-stimulation. And this is before people even knew that what the ligand was, or even that there might be a ligand for CD28. Okay, so that molecule had been found. Okay, so this suggests there's something going on there with this co-stimulation. That's Carl June, as shown as mentioned in the text. I think, yeah. Okay, here's these two separate big slides. So here are a couple co-stimulatory receptors on T cells. CD28 interacts with CD80 or 86, ICOS with ICOS ligand, activation of naive T cells. Co-inhibitory receptors, get to those in a minute. CTLA4 also interacts with CD80 and 86, PD1, ETLA. These are all involved in negative regulation, maintaining tolerance, reducing inflammation, contracting the T cell pool. Okay. So first it was exciting that there were these co-stimulatory receptors needed for optimal activation, and then while looking for things that might bind CD28, they found CTLA4. So our model is that dendritic cells are activated through pattern recognition receptors to make co-stimulatory molecules and make cytokines. Jump through this. So positive co-stim receptors facilitate activation. CD28. Expressed by naive and activated CD4 cells. Made by mouse CD8. So interestingly, not all human CD8 cells have CD28, and people have tried to explain away some things with that, like aging. There's fewer with CD28 with aging, and people have tried to have that as an explanation for a lower immune response with aging. Okay. So with the TCR signal and signaling through CD28 initiates expression of IL-2 and BCL-XL, a survival factor, anti-apoptotic factor. ICOS expressed on memory and effector cells. It's inducible, binds ligand from activated APCs. they may help to maintain activity on already differentiated cells. So the ligands for CD28 are CD80 and CD86. In the older literature called B7-1 and 2, this book says these have different intracellular domains, so they may have some signaling function that's different on the inside of the APC. Only professional APCs make CD80 and 86, and dendritic cells constitutively make them. Macrophages and B cells upregulate. That's one of the reasons for thinking that dendritic cells are essential for initiating responses. So so this interaction now brings in the PI3 kinase pathway, and again, believes, as we explored at the beginning of class, we believe the function here is a check on inappropriate activation, where you need two signals, such as activating the dendritic cell. Again, ICOS through interaction with this ligand provides co-stimulation for already activated effectors. CTLA-4 is a member of the same family as CD28, but is a negative co-stimulator expressed after activation. So it's induced later as if it's a dampener, peaks later. It's expressed at lower levels in CD28, but is a higher affinity, so it seems to be putting the brakes on. PD1, CD279, numbers are way up there, for programmed death 1. So it binds two ligands, so immediate tolerance, a lot of effort with PD1 in cancer treatment now. So early on, they found CTLA-4 as something that bound CD28, and they made knockouts, and one of the early observations that it was a negative effector was that CTLA-4 knockouts had lymphoproliferative disorders, meaning they had a really large number of T cells develop, and again, note the power of knockouts, know what knockouts can do, but also remember, sometimes they have problems. So in the box in your text, it talks about the idea of a checkpoint inhibitor, and I don't know if that's really the best usage, but the idea of using anti-CTLA-4 to block CTLA-4's function, and therefore to help in responses that you want, such as an anti-tumor response. Okay. So this is a great example of applying the basic science to the clinic. And the name associated most with that is James Allison. And we'll talk a little bit more about that later. So the idea of clonal energy, non-responsiveness, provide tolerance, and only signal 1 is received. So this might be if a T cell manages to get through the thymus negative selection. Or also co-inhibitory receptors can also induce energy. So clonal energy if a co-stim signal is absent. So glutaraldehyde fixed cells can't be induced to express CD80 and 86, and that renders T cells unresponsive. Again, a safeguard against autoimmunity. And again, to give the example, prevent setting an attack on islet cells by a rogue T cell that escapes selection if there's not appropriate co-stimulation. So this has been worked out mostly with CD4, but there's some evidence in CD8 cells as well. So anergic cells don't have any effector functions, they can't proliferate or secrete cytokines. And there's some suggestion that some of these anergic T cells may become Treg cells. So now the idea of signal 3 and cytokines that may be present, and again, it's important that these cytokine concentrations, the local concentration is what is important. IL-2 is a very important one. Autocrine and paracrine cytokine. So T cells produce this, and also the receptor for it. Binding produces a very strong proliferation signal during activation. Upon activation, it ups transcription of IL-2 and also CD25, the alpha chain of the IL-2 receptor, so that increases the sensitivity to IL-2. And also activation increases the stability of IL-2 mRNA. Other cytokines are polarizing cytokines. Polarizing meaning that they can direct the T cell down multiple paths with different effectors. So here's going for more of an overview slide here.
Can I ask
Yep.
a question? So the polarizing cytokines is, you said it, so the one cytokine is capable of multiple?
No, that's more for determining, for instance, that this cell will become a Th1 type helper cell
Yeah,
or a Th2.
okay.
So that's what that's meant for. Okay. So here's some comparison of different presenting cells and co-stimulatory properties. Dendritic cells and macrophages are both phagocytic. Receptors for PAMPs and also DAMPs, danger-associated molecular patterns. DCs constitutively express high levels of class 2 in antigen processing machinery, express co-stimulatory molecules. B cells are different in that B cells are really good in a specific case for the cognate antigen, because B cells have a specific receptor, and if they take in that antigen, they not only are stimulated, but they also can process that antigen for presentation to T cells. Let's see. So, different merits of different antigen-presenting cells. There's overlap, but dendritic cells seem to be the most potent. Book notes that the medullary thymic epithelium also expresses class 2 in co-stimulatory molecules, as you would expect for its role in negative selection. Note again the value of dendritic cells as sentinels. B cells important for their cognate interactions. Again, they can then present to T cells and T cells onto the follicle and get T cell help. Now, another, call it almost a side issue here, superantigens, what you heard, which you've no doubt heard of, what makes them super? Superantigens can turn on a wide number of T cells. So they're proteins made by viruses or bacteria that bind to specific V betas outside of the groove. So they interact with the class 2 and the V beta of T cell receptors. So they will interact with all cells bearing a certain V beta. So this can be a really large number of T cells. Some of them interact with more than one type of V beta. So what does this mean? This means a super antigen is super in that it turns on a really large number of T cells. It says effectively short circuits need for co-stimulation and dramatic cytokine secretion, so much so that it's harmful to the host. So I don't know, I'm always looking for analogies, and I think I I think I said before, at least in my micro class, and not here, that it's kind of like everyone calling 911 at the same time from all over Bellevue, or maybe pulling all the fire alarms. Completely strong, evoking a strong response. It's not that useful, and it can actually be so bad for the host that it can lead to toxic shock, as in toxic shock syndrome. So that's what super antigens do. So they don't bind in the groove, activate a lot of T cells. I have written down here in the book that 5% of total T cells may be activated. I thought it could be more, but this includes like the toxic shock syndrome toxin. Yeah.
Can you explain a situation in which this response would be beneficial? Like, why do we have this capability if it's?
Oh, I don't think it's beneficial. The host is
Oh,
beneficial.
it's never beneficial.
Yeah,
So there's
yeah,
okay.
Yeah. I mean, they say it's questionable how it's beneficial even to the pathogen, but I think it is because it just messes up the host response.
Okay.
Yeah. So here are some of the known super antigens. So some enterotoxins, like food poisoning toxins from staph, and these are all the V betas that are activated, that are bound in the mouse. So you get this idea how bad that is. And of course, the V betas are different in mouse and human. So you have this, but the important thing, the toxic shock syndrome toxin, it was associated with super absorbent tampons around 1990 and caused deaths. People want to figure that out. Okay. So a few of these others. Also, these have been used in basic research too, especially before people had the ability to work with transgenic systems where a large number of the T cells had specificity for one protein. People didn't have that. They could work with these super antigens, which would interact, which would delete in the thymus or turn on in the periphery a large number of T cells. So this was kind of a system of choice back then. So yeah, there are endogenous super antigens on integrated retroviruses and also on some pathogens. One group of integrated viruses produces what are called minor lymphocyte antigens, MLS. And again, damage is caused by the massive response of the immune system. The book questions the benefit to the pathogen of super antigens, and I think we kind of understand it because it messes up the immune system so much. Okay, let's start to think a little bit about differentiation now. So now with proper activation by signals 1 and 2, upregulation of survival genes, including BCL2, transcription of IL-2 and IL-2 receptor, activation, robust proliferation, making a lot more cells with the same specificity, and leading to differentiation of appropriate cells. You see that here. Well, the idea of memory versus effector cells. So these activated cells may divide 2 to 3 times a day for 4 to 5 days, so up to 2 to the 15th cells from one. And now, as they do this, they gain functions. So let's think of all the things T cells might do. CD8, CTLs kill virally infected cells, can leave the area and go into circulation. CD4 helpers provide help to macrophages, B cells, and other T cells. Some of these may stay in secondary lymphoid tissues. These effector cells now have a relatively short lifespan, but there are memory cells that live a long time, hence being called memory cells. And of course, it's not well understood what makes some cells become memory and others not, but obviously very important for long responses to vaccines, etc. So our story now is of categorizing these different effectors, these different groups, and but the story is also of people continually, like a lot of things in immunology, once they say it's here like this or like this, continually finding new groups and new ways to divide. So it is recognized for sure, everyone recognizes that CD4 helper T cells are diverse. They can secrete cytokines acting in an autocrine or paracrine way, influencing other cells and influencing themselves. And these CD4 T cells can be divided into distinct subsets, although some of that is even questioned now and coordinate Th1 or Th2 responses. And I think we'll talk about that next time. Okay? So we'll do activation, signal 1, signal 2, and signal 3, and then with CD4s going down different paths that are ideally tailored to the appropriate antigen. And one of the take-homes is that some of the antigen, by what it activates, including what pattern recognition receptors it activates, can steer the response. Okay? Okay, questions about this or anything else? Journal club or test? Well, I'll try to be as available as I can, of course. And yeah, good. Okay, well, see you next time.